Welcome, my digital histology trailblazers. We're going to be talking about the eye. And for those who are joining me for the first time, I'm Dr. Alex Zhurav. I am a veterinary pathologist and a very strong digital pathology enthusiast. And in this series, we're going to be talking about histology, about the normal, so that whenever you need to recognize abnormal, you know what the baseline is. This is the eye, and you can see it's round, and it have different parts. I love how it is explained because we have in the big picture we have like three different tunics, three different layers of the eye. We have the fibrous tunic, the vascular tunic, and the neural tunic, and they go basically from the outside to the inside. This is going to be our nervous tunic. So fibrous, then we have the vascular, and we have the neural tunic. And the key roles of these tunics are going to be, for the fibrous one, is going to be protection and shape. So that's going to be the stuff that's going to, so the fibrous is going to be protection and also giving this eye a shape, this round shape. Here we have like a little bit of an angle. This is going to be the fibrous tunic role. And then, so let's look at it. This is our cornea. And we're going to be diving into it, but I just want to show you that, okay, this is the protection from the outside. It has to be protected. Other than my glasses, I need to have some more protection. It has to be nourished. So for nourishment, we're going to have the vascular level where there are going to be vessels. But the main goal of the eye is to see, right? So we have to have the neural connection to the brain, and this is what we have right here on the back of the eye, and it actually like goes out to the brain because this is our optic nerve. Starting with the fibrous tissue from the very beginning, from the very front, we have the cornea. And cornea has a couple of layers, as you can see here. Okay, we can go even like super high. Well, 100x. We can see everything here, but let's do this and see the structures that we have here. You can clearly see, so the first dot is near the corneal epithelium. And corneal epithelium, also called Bowman's epithelium, has a membrane. Here's the membrane. I don't know why it's doing this. And here is this epithelium. And this is stratified epithelium. This is squamous. These are squamous cell. This is a squamous barrier. And every epithelium is laying on a membrane. And it's not that well visible, but the membrane would be here. This is what it would be. Every epithelium sits on it, and this particular epithelium is called Bowman's epithelium. And then we have a bunch of these pink fibers. This is collagen and fibroblasts arranged in a specific way, a very parallel way that lets the light go through. And this is it's transparent. And this part of the eye also has a membrane. It's called Descemet's membrane. It is simple squamous epithelium, and this Descemet's membrane pumps the fluid out. It has to stay transparent. If there is too much fluid, it's going to get cloudy. We don't want that. So this membrane here, this super thin one, it's called Descemet's membrane. And this is our endothelium because it's the internal epithelium. So this is our cornea. And then cornea has like a sister or continuation that also belongs to this fibrous layer that extends all around the eye here. And this is called sclera. You can see that in the sclera, you also have these fibers, but they're kind of denser. I don't really know if we can appreciate it microscopically. I see that these are a lot more regular, but the difference is that this is not transparent anymore. In the sclera, the fibrous tissue is not transparent, whereas in the cornea it is. So you can see through it, and in the sclera you cannot see through it. So, and then they have this part where they join. This is the transition here, right here. This would be our cornea, and this is our sclera. And this transition from one to the other is called limbus. We have the ordered collagen, and this is supposed to be a little bit less ordered. Honestly, to me, it's ordered as well, but maybe somehow it is not. But obviously, we have other stuff here on this limbus, on this border. We have all this stuff, and this particular thing is iris, and this thing here is ciliary body. Here, no, actually, it extends here. Ciliary body with ciliary processes. And let me show it to you from a little bit more of a distance. You can see all these landmarks at low magnification. It's telling us it's 0. I don't think this is accurate. Maybe it is accurate. 0 magnification, that's true. Come on, what's going on? What did we do? Oh, do you see this little thumbnail? Actually, you cannot see it, but I'll show you in a different way. No. Anyway, on the left here, you can see part of it is a little thumbnail. So to go somewhere specific, you can just click on this thumbnail. And this is something typical for digital slides. You usually have this little thumbnail where you can click and it's going to take you to a different part of the slide. But what we were talking about was this limbus. And we are transitioning from the fibrous to the vascular tunic. Here, this is going to be part of the vascular tunic, and vascular tunic is supposed to nourish the eye, is supposed to give first iris. You can see that there is space in between these two flaps, these two pieces of the iris, and we have the anterior border of the iris and the posterior epithelium. So let me show it to you from closer. This is the anterior and side, and this is the posterior side, and we're going to be talking about what's there as well. But it's dark. You can see that it's dark. It's melanin rich. It's pigmented, right? And we're going to be talking about this pigmented epithelium. It extends also to the ciliary body, all the stuff that is dark, and it's called retinal pigmented epithelium. I mean, it's continuous with the retinal pigmented epithelium, and it's going to go all the way to the back, to the retina, and that is our layer that's going to accept light and translate light into what we see in the eye. But going back to the iris is going to be the thing that gives the color to your eye. So you can see this gap here. We saw this gap already, and it regulates how much light is going into the eye. This is the function of the iris, to either let a lot of light in or let a little bit light in. And we have our ciliary body here, and it also has this dark epithelium, the pigmented epithelium. We have processes of the ciliary body right here, ciliary body processes, and we have some smooth muscles, and The function of this is controlling the shape of the lens. So this is going to be attached to the lens, and this is the lens. Actually, all this is the lens, and the lens focuses light into the retina, and it needs to be clear. And if it's not clear, then we have blurry vision. But basically, the ciliary body attaches to the lens and regulates its shape for focusing, for being able to see. We have also the choroid, which is the vascular layer of the eye. And here, it's gonna be nourishing the eye. And I would love to show you like vessels that have blood in them, but I don't see blood, but I see the vessels. Okay, so here, they're nourishing the eye. Let's go to the back and look at the neural tunic. The neural tunic is on the back, gets light, and translates it into images in the brain. And the main part of this neural tunic is the retina. Here, this is the retina. You can distinguish several different layers of the retina. And I have a table in the notes about all these layers. Let me show them. Let me show the table to you. Honestly, you probably, if you have like an exam or something, you will need to learn them. I never had to actually, like in real life, talk about them. So, like from the histology standpoint, you can have the RPE, the retinal pigmented epithelium. So this is going to be the thing here. Sorry. Can I share two screens? No, I cannot. Retinal pigmented epithelium, photoreceptor layer, external limiting membrane, outer nuclear layer, outer plexiform layer, inner nuclear layer, inner plexiform layer, ganglion cell layer, nerve fiber layer, and internal limiting membrane. So a lot of membranes, I have to say. And now let's look at them on the image itself. Okay, so we're starting from the retinal pigmented epithelium that we already showed, that goes like all around the eye. Here are my limiting annotation capabilities. So all this dark thing going around the eye is our retinal pigmented epithelium, also abbreviated as RPE. And this is where we are going to be counting the layers from. So let's see if I can even count 10. We have the RP 1, 2, 3, 4, 5, 6, 7. Okay, I see 7. So, but there are like membranes. Maybe 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. Okay, so these membranes count as well. So yeah, I guess I need to do a separate live stream on retina and go super deep in. But you can see what we have here. So we have the retinal. Let's see if I can make dots here. Retinal pigmented epithelium. And then we have rods and cones in the photoreceptor layer. And then we have the membrane, we have the outer nuclear layer. So these nuclei is the outer nuclear layer. Then we have outer plexiform layer, inner nuclear layer. So these are like super visible from the outside. Inner plexiform layer and ganglion cell layer, and they all have different functions. But the signals going to the brain go through here, through optic nerve. This is optic nerve, and it goes to the brain. And that was our eye. Let me know if you are interested in the notes from this, with all the layers of the retinal epithelium and everything we discussed on the eye. And something else I wanted to show you. So if you're interested, if you go to conferences, if you want to show fancy in the lab, or and have people ask you, "Oh, nice earrings. What is it there?" We have ones that are with a multinucleated giant cell. Does it focus on it? This is a multinucleated giant cell, a type of immune cell, macrophage, actually, a bunch of macrophages that come together. We have one with cartilage. Come on. Okay. And we have one that is not unpacked yet, with a special stain, Alcyon blue of colonic crypts. So if you want to look different and show what you look at under the microscope, you can get them in our store. The QR code is on the screen. And also, those slides that we were viewing now were possible because of a technology called digital pathology. This is something I have been promoting for the last 10 years. Digital pathology makes us be able to view glass slides, to view tissue digitally. So if you're interested in learning more about digital pathology, there is a book I have for you. This book is absolutely free. You can grab it also by scanning this QR code, and then you will learn more about digital pathology. And we're going to be updating this with a new chapter on AI very soon. So if you grab the version that I have available today, you will automatically get the updated version when I'm ready with the updates. If you're watching the replay, let me know in the chat if you have any questions. What would you like to learn more about? And in the future, meaning probably already next week, we will be talking about mouse histology and rat histology. But if you're interested in human histology, don't worry, the tissues look almost the same. Like, you know, the histology of a rat liver, you will understand the histology of human liver. So this is totally cross-disciplinary. So if you are a researcher that needs information on animal tissue, that's going to be perfect. But if you just want to brush off or step up your histology game, it doesn't matter the species, because in mammals, these appearances, the tissue appearance translates. And me, as a veterinary pathologist, I can totally recognize all the human tissues and actually describe their structures based on what I learned in veterinary histology. So no worries about that. Uh, Thank you so much for joining me. If you're interested in the notes, let me know in the chat. Just put notes, and I'm going to reach out to you with the notes from today's class. Thank you so much, and I talk to you in the next episode.